Hi, my name is Hannah Swain, and today I'll be sharing with you part of my first chapter of my dissertation. Now, first, let's travel to a beautiful reef off the coast of Texas, known as the Flower Garden Banks National Marine Sanctuary. Now, we're about 100 miles off coast here, and deep down in the ocean, about 60 to 80 feet, we have a gorgeous coral reef. On this coral reef, because of its remoteness and isolation, it has very low prevalence of coral diseases. However, in September of 2022, unfortunately, there was a disease outbreak. We were able to go and collect samples from one brain coral known as Pseudoploria strigosa. And we take a unique sampling scheme where we collect three different types of coral samples across the species. We have this healthy portion from a healthy colony. We have a diseased portion right on that disease line of the diseased colony. And then we have the HD portion, which is a portion on the diseased colony that still appears to be healthy. And what I do with my research is look at the gene expression of both the coral and the algal symbiont that lives within the coral's tissue across these three sample types, or as I'll be referring to them as health states. I'm able to get my gene expression data through RNA sequencing and transcriptome assembly. And what we are doing within our large collaborative group is trying to characterize this coral disease. And we're doing this by looking at all of the members that live within the coral holobiont. We have, just to name a few, some viral and microbial diversity. We, of course, have the host and symbiont gene expression. And lastly, we have histopathology of the coral's tissue. Now, today I'll be focusing on the gene expression and the histopathology by looking at two traits within the histopathology work. We have the average symbiont area, which looks at the algal symbiont itself, and second, the average vacuole area, which is the vacuole that houses that algal symbiont. Now, in response to this unidentified disease, both the symbiont and the vacuole shrink. So I wanted to see what types of genes are correlated to the shrinkage of both the average symbiont area and the average vacuole area. And what I see is that in response to the disease, the coral and both the algal symbiont are upregulating genes that are linked to an innate immune response, apoptosis, so programmed cell death, and even protein degradation in response to the disease. Now I see similar types of genes as well upregulated in correlation to the shrinkage of the vacuole too. Now, what this tells me is that the diseased colony, both the coral host and the algal symbiont, are having really strong responses to this unidentified disease. And it seems as though there is a loss of symbiosis from the coral host to the algal symbiont, or the algal symbiont is beginning to either degrade from potentially nutrient starvation, or it's preparing to be expelled from the coral itself. Now, luckily, an interesting find is that we went to this coral reef 6 months later, and most of these disease colonies actually had recovered. So another focus of my research is specifically looking at this HD portion of the colony to see what type of response is this side of the colony mounting, and is it strong enough to halt the disease from spreading? Thank you very much for listening, and I hope to see you at Discover Symposium later in April.